Hello, can people hear me?
We can hear you.
Gonna assume yes. Good morning, everyone. Today we have Professor Amir Ghaem-Maghami from University of Nottingham, United Kingdom, giving a talk on immuno-engineering. Professor Ghaem-Maghami is an old friend. He's been a friend of ours for about a decade and been working with us, publishing great papers. He's a professor of immunology and immunobioengineering at the Faculty of Medicine at University of Nottingham, UK. He has an MD and a PhD in immunology from University of Nottingham. He has done a postdoctoral work in Leicester and Nottingham, and eventually become a lecturer in immunology, followed by his promotion to associate and full prof at University of Nottingham. He works on immuno-instructive biomaterials. He has won many awards. He's a fellow of Royal Society of Biology and UK Higher Education Academy, and serves in other editorial and advisory boards. With that, I give the floor to Amir.
All right, thank you very much, Mehmet. Can you hear me okay? All right, so I'm assuming you can hear me.
Yeah, you can.
All right, great. So as Mehmet said, I'm going to talk to you about immune-instructive materials, an area that we have started working on in the last couple of years, and give you a number of examples of the potential applications as we move forward. So for some of you, I know that have been to Nottingham in our labs, so this is just a view of our campus, and I think if you look really hard, you can see in the corner, top corner, right corner of the picture on the right, where our lab is located. So our group called Immunology and Immunobioengineering, and historically what we have focused on is host-environment interaction. So we are interested to understand how different environmental insults interact with the body and with the immune system, and what are the consequences of those interactions, how those interactions shape the immunity, and leads to either protective or deleterious immune responses. And in last couple of years, as I said, in extending that notion of host-environment interaction, we have been working on how biomaterials interact with the immune system, which is the area I'm going to talk to you. But in parallel, we also do some work on immune-competent tissue models, where we try to incorporate immune cells within tissue models, particularly barrier tissues, so that we have biomimetic platforms that help us to better understand how these environmental insults interact with the immune system. But today I just focus on this immune-instructive materials concept. When we talk about the immune system, I mean, from the sort of evolutionary point of view, the immune system has evolved to protect us against insults. Pathogens come to mind first, and the main job of the immune system is to deal with pathogens and keep the body and the host safe. But at the same time, immune system also targets other things within the body, including tumors. So the immune system plays an important role in trying to deal with tumors and cancers, of course, not always successful. But there are other instances that the activation of the immune system is not necessarily helpful. For example, when the immune system starts targeting self-tissues in the context of autoimmune diseases, that's always harming the host. But the other set of scenarios where the immune system activation is not necessarily helpful for the host is where the immune system identifies a foreign material, trying to protect the body from that foreign material, but the consequences of that could be harmful to the body. So this could be in the context of materials that are entered the body incidentally or accidentally, left inside the body after surgery, or it could be things that we introduce to the body for therapeutic purposes in the context of regenerative medicine or medical devices. The other function of the immune system, which has emerged in the last few years a lot stronger, is the contribution of the immune cells in the process of healing and tissue regeneration. So we know that without the contribution of different immune cells, the healing process would be interrupted. So basically, there are lots of evidence from depletion of various immune cells or targeting them shows that they're absolutely necessary for orchestrating the healing processes. And they have a very close interaction with stromal cells that are also important in healing. So that's another aspect of what the immune system does. Now, when you have tissue damage, the immune system gets activated, and you have some acute inflammation, which is necessary, as I said, for the process of healing. But there are scenarios where this acute inflammation doesn't lead to healing and resolution of inflammation and leads to chronic inflammation, and that chronic inflammation leads to fibrosis and loss of function and tissue damage. Some of these scenarios that underpin this pathway of chronic inflammation and fibrosis include persistent infection, extensive tissue damage and tissue loss, poor circulation, or existence of a foreign body that the immune cells cannot deal with. And that's where the sort of concept of foreign materials becomes relevant. So as I said a minute ago, foreign materials could end up inside the body, not intentionally, but we also have increasing number of instances that as part of a medical intervention, we introduce foreign materials to the body, and implants are probably the best example of that. And there is an increasing now use of biomaterials in different branches of medicine, from acellular scaffolds that are used in tissue engineering and regenerative medicine, to implantable cancer vaccines, where using biomaterials, you try to promote a specific immune cell activation, adjuvants for vaccination, or more sort of conventional medical devices that have been used for many, many years, like your hip and knee implants, dental implants. So all of them are introduced as part of a medical intervention, and the immune system responds to these and sees them as foreign bodies as well. And these interactions lead to activation of the immune system. So, and the consequences of these could be detrimental, and some of the major impediments to the successful integration or successful application of medical devices are things that are a consequence of adverse immune responses. And those include chronic inflammation, fibrosis, and also non-functional immune responses at the site of a foreign body could contribute to infection. And collectively, around 20% of people who receive implants, medical devices, and biomaterials for therapeutic applications suffer from one of these complications that, as I said, they arise from adverse immune responses. So historically, to circumvent this problem, people have tried to come up with stealth biomaterials, biomaterials that cannot be seen by the immune system. But we know that for even the best immune competent systems or biomaterials can still interact with the immune system and cause chronic inflammation, leading to the complications that I mentioned. So that's why we and others have been trying to come up with this concept of immune-instructive materials. So these are materials that have inherent properties that can leverage on body's own defense mechanisms and regenerative capacity to not only not be seen by the immune system, but through active engaging of the immune cells, They can reduce inflammation in situ, promote regeneration, and reduce risk of infection. And of course, when we talk about the immune system, you have a very complex network of different cells, mediators, proteins. And for the purpose of this talk, I will focus on two main innate immune cells that are macrophages and dendritic cells. And the reason they are important, because there is plenty of evidence, both experimentally and from the human studies, that these cells are very important in orchestrating downstream immune responses, and particularly orchestrating the response to foreign materials and foreign bodies in general. And also they influence and they shape adaptive immune responses. So I focus on macrophages and dendritic cells in the context of their interaction with biomaterials and foreign bodies, basically. So macrophages, I'm sure most of you, if not all of you, have heard about them. So these are phagocytic cells that can monitor the environment, deal with foreign materials, primarily pathogens, but anything else that is foreign, they can sense it, sample it, and then try to deal with it accordingly, according to the nature of what it is. For simplification, people have historically divided these cells into two classes, it's called so-called M1 and M2. The M1 is representative of a class of pro-inflammatory macrophages that have very important functions in dealing with infection and also tissue injury and resolution of infection. But an uncontrolled activation of M1 could have serious consequences in terms of tissue injury. And then the M2 macrophages that are representing the anti-inflammatory phenotype or end of the spectrum, they are important in tissue repair and tissue remodeling, but excessive activation of M2 phenotype could on its own have some side effects, including promotion of fibrosis. And eventually what you want to happen is a balance between these two phenotypes. That's one point. And the other point is that although we talk about M1 and M2, it's important to bear in mind that these cells in the body, they have a spectrum of phenotypes that is determined by their environment. So they're very responsive to changes in the environment, they're very responsive to microenvironmental cues, and they can change their phenotype accordingly, and they're very plastic. And this provides many opportunities for changing their phenotype by controlling these environmental cues. And I'm sure most of you are familiar with different approaches that are available to influence the environment. You can use soluble factors, cytokines, you can use other cells to produce soluble factors, but you can also modulate the physicochemical properties of a substrate, and through that influence the cell phenotype. And in the rest of my talk, I will focus on some of the work we've done in leveraging on this plasticity of these cells, and also our ability to fine-tune the physicochemical properties of substrates to influence the functional properties of these cells. A number of years ago, when we started asking the question of whether the surface chemistry could influence macrophage phenotype, we decided to modify the surface of a tissue culture polystyrene using plasma etching to simply change the wettability. Just as a readout, obviously, that will also have other influences on the surface properties. But by doing that, we were able to generate surfaces that were highly hydrophilic, as opposed to ones that were highly hydrophobic. And that simple change in the surface chemistry would allow us to change the phenotype of monocytes. These are human primary monocytes that are seeded on these different surfaces and allowed to differentiate into macrophages. And from those experiments, what we saw was that the cells that are cultured on hydrophilic surfaces, they acquire this pro-inflammatory phenotype. And here we are looking at the surface phenotype of these cells, and the red color represents this calprotectin, which is a pro-inflammatory marker, and the green dye represents mannose receptor, which is an anti-inflammatory marker. And you can see that cells on this hydrophilic surface, they express higher levels of calprotectin. They have acquired a pro-inflammatory phenotype, and this is quite in contrast with cells on the hydrophobic surfaces, which express high levels of this anti-inflammatory marker mannose receptor and very low levels of the pro-inflammatory marker. So we are able to differentiate these cells, differentially polarize them by simply changing the surface chemistry by using the plasma etching at different levels. So we have characterized these cells a lot more, and actually the surface phenotype also goes with the cytokine profile and other properties. So this gave us motivation to expand this search for other chemistries and looking at a wide range of different chemistries and see how they could influence macrophage polarization. And that's where we started working with Morgan in Nottingham, who had previously done quite a lot of work on using these polymer libraries for looking at bacterial attachment and stem cell differentiation. Relatively straightforward technology where you can create diverse libraries of acrylates and metacrylates, and then look at cell behavior by seeding cells in a semi-high throughput manner. So we wanted to apply this to macrophages in the first instance and identify chemistries or polymers that are actually able to influence macrophage polarization. So the workflow of this is we separate monocytes from peripheral blood, and these are the precursors of macrophages. So they're seeded on these polymer arrays, and then we use microscopy to initially look at cell attachment, and then following that with more characterization of the cells. But even at the level of cell attachment, you can see that there is a diverse cell attachment potential, starting from polymers that only attract a handful of cells to those that you have hundreds of cells. And that on its own could have major implications in terms of how this particular polymer will interact with the immune system when they are implanted. But we wanted to focus more at that point on the ability of these polymers on macrophage polarization. So using this first generation of arrays, we selected a number of spots that were highly attractive for monocyte attachment, and were able to induce polarization towards either pro or anti-inflammatory phenotypes, and generated a second generation of copolymers, and then screen them again for the ability for cell attachment and the ability for inducing pro or anti-inflammatory macrophage polarization. So this is just to give you an idea about the distribution of these different polymers. You can see that there are polymers that are able to promote macrophage polarization towards highly pro-inflammatory phenotype, again represented by expression of calprotectin as a pro-inflammatory marker in red, and mannose receptor as an anti-inflammatory marker in green. So we had now a number of polymers that could differentially attract these cells, but also induce the polarization towards pro or anti-inflammatory phenotypes in the absence of any exogenous cytokines. And that's what we typically use to polarize these cells. We add cytokines, cocktails to push these cells towards either pro or anti-inflammatory phenotype. But then with these polymers, we're able to achieve that in the absence of adding any exogenous cytokines. So the two questions that were followed after those observations were, what is the molecular basis? So how these polymers are able to influence these monocytes macrophages behavior. And then the second question was whether in vivo these polymers could have any influence on this foreign body response process. And I just show you a couple of slides going through some of that data where we're trying to answer those questions. So one of the things that historically Nottingham is very well set up to do, and particularly in Morgan's lab, they do a lot of surface characterization. And this is the sort of workflow. So you select your polymers of interest from the microarray, polymer array format. You scale them up, and then you subject them to a number of surface characterization analytical tools like XPS and TOFSIMS and so on. And for a number of our hit polymers, the polymers that were able to promote either pro or anti-inflammatory responses, we looked at the bio-adsorbate layer on the surface of these polymers, because our working hypothesis was that the reason these polymers are able to influence differentially cell behavior is because they absorb different proteins from the media at different levels and different combinations of these proteins on the surface. And that's how cells respond to these different bioabsorbates, and they acquire different phenotype. And here it just shows you the XPS analysis of 6 of these polymers. The first 2 are 2 of our pro-inflammatory polymers. And you can see that the thickness of the protein layer seems to be consistently higher than everything else. But if you look at using proteomics on the actual nature of these proteins, you can see that although there are some common proteins present on both M1, M2, and naive macrophage inducing polymers, we have also identified a number of unique polymers that are present on these different groups of polymers. And that goes some way to explain probably why they have the ability of differentially promoting differential polarization of these cells. Of course, we still need a lot more to do in terms of understanding the mechanism, but that doesn't stop us from exploring the sort of translational value of these by using them in vivo, and that's exactly what we have done. So we've used a sort of in vivo subcutaneous foreign body model by coating pieces of medical grade catheters and then subcutaneously implanting them in an animal model, following it up to 4 weeks, and then characterizing the immune response locally, and then the impact on fibrotic capsule formation and cell infiltration, and some other readouts that tell us a bit more about how these polymers could influence cellular responses upon implantation. So in this slide, I just show you some histology data. If you look at the bottom corner on the right, you see a sort of histology of skin, and you see the pieces of subcutaneous fat. You have the connective tissue, and in the top left corner, this is a tissue response to an uncoated polymer. So you have a very typical inflammatory response, lots of immune cell infiltration. So you have some fibroblasts and fibrotic showing fibrotic behavior. And even to an untrained eye, you can see that these responses are quite different amongst these polymers. For example, for the 2 hit polymers that are pro-inflammatory, you can see that the level of cell infiltration is pretty much similar to what we have for uncoated. And for the 2 polymers that are M2 polymers, the 255 and 301, you can see that there are far fewer cells that have infiltrated the immediate vicinity to the implanted catheter. And then the 2 polymers that represent naive macrophages. So these are polymers that don't influence macrophage polarization status. They're highly attractive for these cells, but they don't influence the phenotypes. Basically, they maintain a neutral phenotype, a naive phenotype. So there is a modest cell infiltration, but if you look at the collagen deposition and fibrosis in the next slide, you can see that it becomes even more interesting. If you look at the uncoated, so you have fair amounts of fibrosis and collagen deposition. It's a lot more for the two M1 polymers, similar levels for the M2 polymers, but what's really interesting is that in the M0 and the two M0 polymers, we have substantially less collagen deposition and fibrosis. And based on this, what we hypothesized was that maintaining the macrophage phenotype at a naive state actually could contribute to better resolution of inflammation. And actually that is in line with what we know about the homeostasis, where naive macrophages actually contribute and maintain the homeostasis. But the bottom line is that with these different polymers, we can induce differentially a state of polarization that reflects in the tissue response, collagen deposition, cell infiltration, and fibrotic capsule formation. The other element of wound healing I mentioned very briefly are stromal cells. And as I said, there is very close collaboration between particularly macrophages and stromal cells when it comes to wound healing and wound remodeling. And that's why in parallel, we've had a lot of interest in how fibroblasts behave and respond to these different surfaces, the different surface chemistries. And this is the work that Arsalan, one of our PhD students, have been doing in the last couple of years, having the same workflow, but his cell of interest are fibroblasts, and he's interested in a number of cellular behaviors. One of them is attachment and proliferation, but also fibrotic phenotype of these cells, and using alpha-smooth muscle expression as a surrogate for fibrotic behavior. And through that exercise, what he has identified are a number of polymers that actually repel fibroblast attachment. So you can see that from this graph that there are some polymers that there is very little interest from fibroblasts in terms of cell attachment. And then at the other side of the spectrum, you have polymers that are highly attractive for these fibroblasts. And it's not just the cell attachment, but they also influence cell proliferation and the fibrotic behavior of these cells. And based on these readouts, we have been able to select a number of these polymers that promote fibroblast proliferation, but at the same time, they have this anti-fibrotic potential. And it becomes very interesting when we add TGF-beta, which promotes fibrotic behavior. And for these anti-fibrotic polymers, even in the presence of TGF-beta, we are able to suppress the fibrotic behavior. So now what we are thinking is that the combination of these two could actually influence this whole process of fibrotic capsule formation, collagen deposition, and could influence the foreign body response against implanted materials. But at the same time, they have other potential applications, for example, wound healing process, which is heavily reliant on both fibroblasts and macrophage behavior. And these are the things that at the moment we are exploring both in vitro, but also using animal models to better understand how we can influence the wound healing process and how we can influence the foreign body response against implants. So at this point, I just change gears and talk about a little bit dendritic cells. As I said, dendritic cells are professional antigen-presenting cells, and they act as a bridge between innate and adaptive immune systems. So they can instruct the adaptive arm of the immune system and shape the T cell polarization that are most important effector cells when it comes to dealing with different environmental insults. So one way of influencing adaptive immunity would be through influencing dendritic cell phenotype. And dendritic cells do that by expression of various surface markers, and the maturation status of dendritic cells is highly important in the way in which they crosstalk with T cells. So if we have tools to influence dendritic cell phenotype, then we are able to impact T cell polarization and therefore adaptive immune responses. And that's why we were interested in finding polymers that are able to influence dendritic cell behavior. And then the phenotype, the functional phenotype of dendritic cells determines the way in which they influence T cell polarization. So they can induce T cell energy, they can induce very robust T cell responses that would be beneficial for dealing with infections, dealing with tumors and cancerous cells. And at the same time, if you can induce tolerogenic dendritic cells, they are able to then induce regulatory T cells, which will be important in dealing with autoimmune diseases, for example. So the workflow for identifying polymers able to influence dendritic cell behavior, very similar to what we've done with the other two cell types, except that in this case, dendritic cells are not adherent. So therefore, we cannot use that array format. So that's why Lisa, as part of her PhD work, has been creating these polymer libraries within tissue culture wells, so that she can seed cells inside the wells. They have their own defined media, and then we can look at the cell behavior, phenotype, cytokine profile, and then have an understanding of how cells have responded to different polymers. So this is the workflow for that. And through this exercise, we have identified a number of polymers that support the viability of dendritic cells. In some cases, much better even than tissue culture plastic. And for the ones that had good viability, we looked at their impact on dendritic cell phenotype in the first instance by looking at maturation markers. And I here just show you a couple of examples of what we call stimulatory polymers. So these are polymers that induce dendritic cell maturation. So just to give you an idea about how potent they are. So if you look at the data from the LPS, so this is lipopolysaccharide, an endotoxin from gram-negative bacterial cell wall, and this is the most potent inducer of dendritic cell maturation. And here you have around 100% of cells expressing this particular marker, CD83, which is a maturation marker. Same for this other marker, CD86. So this is what we can achieve with this endotoxin. And if you look at this selection of stimulatory polymers, you can see that they can induce the maturation of these cells almost as good as LPS. And then on the other side, again, if we use these LPS-induced stimulated cells as our sort of controls, you can see that, again, we can achieve near 100% maturation, but then these inhibitory polymers, and even in the presence of LPS, they maintain this immature status of these cells, of dendritic cells, which could be highly valuable if your interest is promoting energy and regulatory T cells. But I say a little bit more about the stimulatory dendritic cells, because that's where we are pushing this work forward in terms of the ability they have to promote T cell polarization and activation. And to understand that in vitro, what we do a lot actually in the lab is to set up co-cultures between dendritic cells and T cells to simulate what happens in the body, because this is exactly what happens in the body. You have dendritic cells in the peripheral tissue, they interact with a stimulant, and then they migrate to lymph nodes where they crosstalk with T cells. They transfer all the information from what they have seen in terms of stimulation, and then they support the activation of that T cell, which is determined by the maturation status of this dendritic cell. So we can set up these co-cultures and then see what is the impact of these polymer primed dendritic cells on T cell proliferation and T cell activation. I just show you some data on interferon gamma production by T cells that have been co-cultured with either stimulatory polymers or inhibitory polymers. Interferon gamma, for those of you who probably don't know, is a signature pro-inflammatory cytokine. So very important in lots of pro-inflammatory events like dealing with pathogens or dealing with tumor cells. And here you can see that if you look at interferon gamma production by T cells that have been co-cultured by polymer primed dendritic cells, stimulatory polymer primed dendritic cells, you can see that the level of interferon gamma production here, shown in green, is very much similar to what we can achieve by stimulating these T cells with this anti-CD3, which is a sort of mitogen for T cells. So you can activate T cells, all T cells, and then they produce maximum amount of interferon gamma. So what we can achieve becomes very close to what can be done by anti-CD3 stimulation. And then the opposite is what we see with this inhibitory polymer. And you can see that T cells that have been co-cultured with inhibitory polymer primed dendritic cells, the level of interferon gamma production is very similar to the control, to dendritic cells that haven't been stimulated at all. So this is quite exciting, but one specific application that we have in mind is to understand how then these stimulatory polymers are able to influence the way that T cells interact with tumor cells. So the specific T cell that deals with tumor cells are CD8 T cells, and the workflow for this is a little bit complicated, but I try to explain it very quickly. So what we do, we get dendritic cells from a donor. We prime these dendritic cells on these polymer surfaces, in this case, two of the stimulatory polymers, and then we feed these dendritic cells by this MCF7 tumor cells, the breast cancer tumor cell that has been lysed. So these cells then present the antigens that are specific to this tumor cell on the surface. They then we set up co-cultures of these dendritic cells with naive CD8 T cells from the same donor. So these dendritic cells that are now conditioned or primed, they pass on that information and these antigens to the CD8 T cells. Then you co-culture these now tumor-specific cytotoxic T cells with tumor cells, and then you look at the viability of tumor cells after those co-cultures. And this is just the snapshot of the results. At the top left, you have tumor cells with dendritic cells that with T cells that have been co-cultured with dendritic cells that were not stimulated at all. So this is your sort of negative control. So you have the odd dead tumor cell, and the green is obviously representing live cells, red representing dead cells, and it's just a live dead assay. And then for the two stimulatory polymers, you can see that there is much higher green, so there's lots of more dead tumor cells. And if you quantify that, you can see that for both of the stimulatory polymers, you can achieve much higher levels of tumor killing and tumor lysis. And this gets very close to what can be achieved using some more chemical induced dendritic cells and DC-based vaccines in terms of percentage of killing. So this is quite encouraging, and it's something that now we are exploring how this could be translated. We have prepared 3D porous scaffolds from these polymers as well as particles, and we are in the process of building up on the sort of understanding of how they influence dendritic cell behavior and how they could be used in the context of both dealing with autoimmune diseases in a sort of antigen-specific way, but also in sort of tumor killing and DC-based cancer vaccination strategies. So I only have a couple of more minutes, and then I will wrap up. I just want to move on to another line of work that we do in the lab. I don't think a couple of minutes will do justice to it, but in the interest of time, I try to be brief. We talked about chemical cues, but we also know that physical properties and topography, particularly of the substrate, is important in influencing cell behavior. And again, that's the sort of angle we've taken by focusing on the impact of topographical cues on immune cell polarization, using the platform that I'm sure many of you have heard of, the Topo chip, initially described by Jan de Boer in the context of MSC cell differentiation. So these are a collection of primitives put together randomly using a mathematical algorithm, creating features that then are put together on a Topo chip consisting around 2,000 unique topographies. And what we really wanted to understand was how these topographies influence monocytes and macrophage cell attachment and polarization. And again, you can see that we have identified a range of topographies that are highly pro or anti-attachment for monocytes. And not only that, they also influence a spectrum of different cell phenotypes from M0, naive macrophages, to highly pro-inflammatory or highly anti-inflammatory phenotypes. So Matt, who's led on this work, has done a lot of sort of structure-function relationship studies, and we now know that the characteristics of these topographies that influence cell behavior, particularly attachment, now this has been pushed much further in terms of the impact of these topographies on both bacterial, pathogen, and foreign body reactions. Just very quick slide to show you that when these either pro or anti-attachment topographies are implanted subcutaneously in animals which have been impregnated with Pseudomonas aeruginosa, you can see that the pro-attachment cells are pro-attachment topographies attract a lot more immune cells, but also if you look at the mass of the bacteria and you look at the superimposition of the two images, you can see that the pro-attachment properties of these topographies are maintained in vivo, and they match what we have observed in vivo. So for the anti-attachment, you barely have any of these immune cells seen or attracted to these topographies. So again, this is ongoing work, and we think it's quite exciting to show that these micro topographies are actually able to influence cell behavior and microbial attachment behavior in both in vitro and in vivo. So with that, I try to just finish and wrap up everything, and just thank all the collaborators. Lots of people obviously have done the work. Hassan has done together with Leanne, and many others have pushed forward the polymer array work for a couple of years, and Matt, Lawrence, and a few others have contributed to the topography work. And as I said, Lisa and Arsalan have worked on the dendritic cell work and on the fibroblast work and all the sort of material work. We've collaborated with Morgan for the last few years now, and lots of other people have contributed, and also thank to lots of funding bodies who have supported us for doing this. So with that, I just finish and happy to answer any questions. Thank you.
Thank you, Amir, for this nice, informative, fantastic talk. For the listeners, please leave a comment either in the chat box or the participant line. Amir, there's one question I'm reading. What chemical structure is favorable to M2 or has no involvement in macrophage polarization?
Right, so I think the best thing is, so there are pages and pages of description of the chemical structures. So probably it's most efficient if I refer you to that paper published, I think in May, end of May or beginning of June in Matter, where we have described all the polymers that are either pro or anti-inflammatory. And actually, there is also some detailed discussion around the structure-function relationship between the chemical structures and the way they influence cell behavior. So it's all published there. If you don't mind looking at it, it would be a lot more efficient. And it's Rostam et al., and it was published in Matter in June.
All right. Another question, have you checked caspases during T-cell activation?
No, I mean, that's a very good point. We haven't looked at caspases for the T-cell activation. So the main readouts we have focused on so far are cytokine profile of T-cells in these co-cultures, and then the ability of the CD8 T-cells in the tumor killing assays that are described. But we have lined up quite a lot of other things to look at. But at the moment, we don't have any data on caspases.
Another question is, could you please name a few stimulatory or inhibitory polymers for dendritic cells if possible?
Yeah, I mean, that unfortunately I can't, because I think they're going through a sort of patent application. But hopefully soon we can share the nature of or the identity of those polymers. But for the dendritic cell ones, for the macrophage ones, it's all published.
Okay, another one is, are you investigating surface chemistry topography impact on macrophage polarization in any specific application? So
well, there are a number of applications. I mentioned medical devices. But I appreciate that's quite broad. But one specific application that we are now actively following up in collaboration with the Smith and Nephew, who are one of the big players in wound care, is using those polymers in the context of wound care. So that's one specific application. But I think anywhere that you have foreign body response as a problem, which is almost applicable to all medical devices, then you can think of how reducing foreign body response, collagen deposition, fibrosis, capsule formation could be beneficial. I mean, glucose sensors is one that there has been a lot of interest in that area. I mean, this is something that we are quite keen to explore, because obviously fibrotic capsule formation around any sensor will render that sensor non-functional because it separates it from the immediate environment. And then dental implants is another application we are actively looking at, because aseptic bone resorption as a result of chronic inflammation is still a major problem in any bone interfacing device, including dental implants. And that is another area we are actively looking at.
All right. I have a question regarding the bio-instructive polymers for wound healing. does making things in 3D make sense, or is 2D sufficient?
Well, I think it depends on the type of wound really you're trying to deal with. So certainly for deep wounds, doing things in 3D could be beneficial. I mean, one thing I didn't talk about is the work we're doing on 3D shapes and the structures and architectures, if you like, and how they influence cell behavior. I think depending on the type of wound, having things in 3D could be certainly an advantage.
Another one, follow up to the surface chemistry on macrophage polarization. They're asking, can this be used for supporting tissue regeneration, such as peripheral nerve regeneration?
Well, I think generally, if you think about regeneration, I cannot think of any scenario that there is no contribution from macrophages in particular. As I said, they're integral in setting the scene for successful regeneration. So on that basis, I would say yes, because when you have regeneration, of course, there are a number of things that need to happen, including vascularization, blood supply, and the contribution from stromal cells. But certainly macrophages orchestrate a lot of those events. And if you can control the macrophage phenotype to promote them towards this pro-healing phenotype, then they could certainly contribute to the tissue regeneration. I mean, bone is another very good example. Bone healing and remodeling, which is as important as healing itself, that there is a very strong contribution from macrophages in that process.
And then follow-up is, which kind of surfaces can be used for this means?
So I think in terms of coating surfaces with these polymers, I don't think there is an actual limit. So, I mean, we have done, and that there is a lot of experience in Nottingham in coating, for example, silicone type substrates. As you here just mentioned, that one of the applications came out of that bacterial attachment screen, which I think was the first one in Morgan's group. Has now been translated as coatings for urinary catheters, which is going now through some clinical trials. So there is a lot of experience in coating that type of substrate. There is a lot of experience in coating metal substrates. But I think in theory, almost any substrate could be readily coated with these. Of course, in some occasions, there is a need for some optimization, but I think a variety of substrates could be coated with these polymers.
Okay. Any other questions from the audience? If not, I'd like to conclude the talk. Again, Amir, thank you so much for this fantastic talk, and hope to see you soon in future events.
All right, great. Thank you for organizing it, and hope you have the rest of good today.